home, staying safe. So in the series of CSIR questions, today we will be seeing the 16th question. So this is with respect to molecular biology. Which one of the following statements related to transcription and processing of mRNA is incorrect? Okay, so you know that the transcription process, in protein synthesis, it takes place inside the nucleus in the case of eukaryotes, and of course in prokaryotes, it is along the DNA in the cytoplasm itself. Both transcription and translation can take place simultaneously, but in the case of eukaryotes, they are spatially and temporally separated. That is, transcription takes place in nucleus and translation in the cytoplasm, in the ribosomes in the cytoplasm, isn't it? Okay, so the option, which of the following is incorrect? First option, during prokaryotic transcription, DNA binding properties of RNA polymerase are altered by sigma factor. So let us see what happens during prokaryotic transcription. If you see here in this diagram, see, this transcription process is carried out by an enzyme, that it is carried out by RNA polymerase, and there is only one type of RNA polymerase you find in prokaryotes. So this has to go for initiation, elongation and termination, all the three. So therefore for this initiation process, sigma factor is required. So these both together, they act as an holoenzyme. And you know that RNA polymerase will attach to the promoter. So take, for example, the gene structure. So there will be operator, promoter, and the structural genes. So it goes and attaches itself to the promoter. So therefore, once this goes and attaches to the promoter region, then see, it will start the transcription process. So the sigma factor is a protein needed for initiation of transcription in bacteria. Okay, and it is the bacterial transcription initiation factor that enables specific binding of RNA polymerase to promoters. Only if it attaches, it will modify in such a way that it will go and attach to the promoter. So therefore this is correct. Is the second one. In eukaryotic transcription, synthesis of rRNA, mRNA and small RNA, snRNA occurs by RNA polymerase 1, 2 and 3 respectively. That is also, it also seems to be true, because in the case of eukaryotes, there are 3 polymerases, isn't it? There you saw only one type of polymerase. Here there are 3 types of polymerase. RNA polymerase 1 transcribes rRNA, and these are the subunits. RNA polymerase 2, it transcribes the hnRNA, heterogeneous nuclear RNA. That is what the pre-mRNA, before processing, isn't it? And RNA polymerase 3, this one, small nuclear RNAs, which are involved in splicing processes, which are important. So they are transcribed by RNA polymerase 3, isn't it? So this seems to be the correct option 2. Okay. Next one, splicing of tRNA. See, you might have heard of splicing of hnRNA, but this is splicing of tRNA. So in splicing of tRNA, so before that we'll see splicing of mRNA. This you might have studied. See, this RNA, when it is getting transcribed in eukaryotes, it has 2 parts. That is introns and exons. Introns are non-coding sequence, exons are coding sequence. So this have to be removed, these introns, so that it becomes a mature messenger RNA. So that is performed by a process called as splicing. So splicing is what? So these structures, these RNAs are removed. How they are removed? These structures are called as lariates, L-A-R-I-A-T-S, lariates, and they are removed by the process called as RNA splicing. So here this occurs in mRNA. In tRNA also, in the anticodon, near the anticodon loop, there will be extra, that is the RNA which has to be removed, that is the intron part. So introns we also find in the case of tRNAs, which has to be removed Okay, so that is removed. It requires two steps. One is phosphodiester bond cleavage, and then second one ligation. Both are required. So both take place. So this also seems to be correct. Now the final one, mRNAs with premature stop codons. The mRNAs are degraded. So when there is something, so when there are premature stop codon, naturally what will happen? This has to be removed. That is done by nonsense mediated decay. That is true. But here mRNAs without stop codon get accumulated. Premature means before actually they have to be stopped. There in between there will be stop codon, so it will not mature. Here in-frame stop codon gets accumulated and translated in the cytoplasm. So here if you see in this diagram, so when there is premature, in the case of non-stop mRNAs, they are degraded and may also trigger translation repression. So here what happens is, so these are degraded. So what happens is, they are not accumulated, they are degraded. Therefore, this seems to be incorrect. So the option which seems to be incorrect is this one. Okay. Thank you. Hope you understood.